I always like to think of the methods section as kind of a cookbook. It's the recipe for my study. And this is a good way of thinking about it, because if you think about a recipe for a nice dish that you're preparing, you want to be able to follow that recipe step by step and get the same result each time. And so this is really an important aspect of the methods. And the difficult bit about the methods is getting that level of detail right, making sure that you have this recipe for your study. So the methods must provide enough information for the reader to understand how the study design addresses the research aims, tests the hypotheses, answers the questions that you've given at the end of the introduction, and of course, whether the methodology and data analyses are appropriate to do that. So is the methodology, are the variables, and are your analyses appropriate for testing your hypotheses? And the recipe aspect of this is reproducibility. So the details that you provide in the methods, the description of your study, should allow the reader to recreate each step. So if someone else were to go out and do the same study at the same location, they should get very, very similar results to you. They should be able to recreate the study the same way that we follow a recipe to recreate a dish. And this is very, very important. So we need to include any details that might influence the way the data are collected or the amount of data collected, the approach we use to analyze the data, so the statistical tests or models, and of course, anything that could influence the results and their interpretation. This is very, very important. So some example of really crucial details are the sampling, a description of the sampling sites and the experimental treatments. And that might include the type, number, and size of the sampling units, obviously the level of replication, and then, of course, the number of samples, the frequency of sample collection, and then other details such as the instrument that you used, the amount of sample that was then used for further analyses, and so on and so forth. And of course, how the data were handled in the data analyses. So these are absolutely crucial details that you really must give in the methods. And you can ask yourself, does the method section really give the reader a mental picture of your study? And could you draw a figure or diagram of the study design or the procedure based on the description in the text? And this idea of creating a picture is quite important, because we must remember we've probably worked on this study for a long time. We're very, very familiar with everything we've done, but we need to describe it for somebody who is seeing this for the first time. So as a reviewer, and sometimes even as an editor, I might read the method section and I cannot understand what the authors have done, and I will try and draw a diagram to see if I can work it out based on the text. If I can't do that, then I immediately say that I can't evaluate the rigor of the science because there isn't enough detail in the methods, or the methods description isn't clear. So this is very, very important, because it is one of the main reasons for rejection post-review, is if the methods are not suited or suitable to test the hypotheses, or if the method's description is so insufficient that the reviewers cannot judge the rigor of the study. So at the very least, you would be asked for major revisions, maybe even reject and resubmit if it's not possible to really understand how the study design answers the research questions or tests the hypotheses. This is very important. So another thing you can also do if you have a complex study is at the end of the method section or at the end of the relevant sections, you can summarize sites, sampling units, treatments, and replicates. You'll often see this as a statement saying, in summary, there were, I don't know, 20 plots with 5 replicants and 4 treatments. Each plot was split into 2 subplots with these subtreatments. And then you have a nice sort of overview of the study design if you have something more complicated. And again, this concept of starting big and finishing small is important in the method section. So if you start, and you'll see this in so many papers, we usually start with a description of the study site, the climate, maybe the soil type or the vegetation type, whatever is obviously relevant to the study, but we start broad. And then we'll build in, for example, transects, plots, sampling units, and treatments. Then we'll start talking about sampling for individual variables. So we tend to do this, start big and build in details or complexity in the methods. And you can really use this to structure the methods and make sure that it's easy to follow what you did and why. It's also very useful if in the methods you refer directly back to your hypotheses. So that you say, to test this, we measured that, or to determine this, we measured that. To actually give the purpose of the methods in each case. That can be very useful for the reader to understand how your study design tests your hypotheses and answers your questions.